Dear students, how are you? Hope you all are fine. We were studying carbohydrates, under which we have already covered monosaccharides. Second type of carbohydrates are oligosaccharides. Oligosaccharides, they are formed by joining of 2 to 6 monosaccharide units, and depending upon the number of monosaccharide units which are joining to form oligosaccharides, they are classified into various categories. First is di means 2. So they are formed by joining of 2 monosaccharide units. Simple example is that of maltose. Maltose is composed of 2 glucose units. Then is lactose, which is formed of glucose and galactose. Then is sucrose, which is formed of glucose and fructose. Trisaccharide, they are formed by joining 3 monosaccharide units. Example is that of raffinose, which contains 1 molecule of fructose, 1 of galactose and 1 of glucose. Tetrasaccharide, they contain 4 monosaccharide unit. Example is stachylose. Penta, as the name itself indicates, they have 5 monosaccharide units. Example is verbascose. Then hexasaccharides, they have 6 monosaccharide units. So depending upon number of monosaccharide units, oligosaccharides, they are classified into different categories. To study how these oligosaccharides are formed, we take the example of a disaccharide maltose, which is formed by joining 2 glucose units. Now here just see, this is pyranose ring of alpha glucose. Alpha because on carbon atom 1, hydrogen is on the upper side, OH group is on the lower side. So this is again a pyranose ring of alpha glucose. So 2 molecules of glucose, they combine through alpha 1,4. Why we say it is alpha 1,4? Because just see, carbon atom 1 of 1 glucose molecule and carbon atom 4 of second glucose molecule, they are participating in the formation of a bond which is formed with the release of water molecule. This particular bond is known as glycosidic bond. So just see, 2 alpha glucose molecules, they join through alpha 1,4. Alpha means, as I have told you, orientation of hydrogen and hydroxyl group. Then the linkage is between carbon atom 1 and carbon atom 4. So it is alpha 1,4 glycosidic linkage. So 2 molecules of glucose, when they combine through alpha 1,4 glycosidic linkage, they form maltose. So this is how a disaccharide is formed. Likewise, in trisaccharide, 1 more molecule of monosaccharide will combine. In tetra, 2 more molecules will combine. So that is how these oligosaccharides, they are formed. Among these oligosaccharides, the most common, these are the disaccharides. Disaccharides, which are also known as double sugars, which I have told you, they are formed by the union of 2 monosaccharide units. There are biological important disaccharides which include maltose, lactose and sugar. First is maltose, which is also known as malt sugar. It is present in the germinating starchy seeds and in malt. So it is composed of 2 glucose units linked by alpha 1,4 glycosidic linkage, as we have studied earlier. Second is lactose, which is known as milk sugar. It is mainly present in mammalian milk. It is formed of 1 beta D glucose unit and 1 beta D galactose unit, which are held by beta 1,4 glycosidic linkage. So now here just see, I have taken beta glucose, because here just see, on carbon atom 1, OH group is on the upper side, hydrogen is on the lower side. So this is galactose molecule. This is again beta form. Beta because OH group is on upper side, hydrogen on the lower side at carbon atom 1. This is galactose. I have told you, glucose and galactose, they differ in the orientation of hydrogen and hydroxyl group at position number 4. So in galactose, OH group is on the upper side, hydrogen is on the lower side. So 1 molecule of beta glucose reacts with 1 molecule of beta galactose molecule. The interaction is again between carbon atom 1 and carbon atom 4. So the linkage is known as beta 1,4 glycosidic linkage, which involves loss of water molecule. So here water molecule is lost and the linkage is formed, which is beta 1,4 glycosidic linkage. So glucose and galactose, they join to form lactose. Third type of disaccharide which is common is sucrose, which is also known as cane or table sugar. This is mainly present in sugarcane and sugar beet. Now it is made up of 1 molecule of alpha glucose and 1 molecule of beta fructose, which are held by alpha 1 beta 4. So means glucose the carbon atom 1 The fructose the carbon atom 4 are they are linked to form glycosidic bond. Now there are two important terms which are generally asked in exams. What are reducing sugars? What are non-reducing sugars? As we have studied earlier, sugars having free aldehyde or free ketone group which can reduce cupric ions to cuprous ions and hence give reaction with Fehling solution and Benedict reagent, they are known as reducing sugars. Examples are glucose, galactose, maltose and lactose. They have free aldehyde or free ketone group. Non-reducing sugar, sugars which do not have free aldehyde or ketone group and hence they do not give reaction with Fehling solution or Benedict's reagent are called non-reducing sugar. Example is that of sucrose. Besides this, there are two more disaccharides. One is trehalose, which is present in insect hemolymph. Hemolymph means the fluid present in the hemocoel body cavity. Second is cellobiose, which is the disaccharide which is formed during hydrolysis or breakdown of cellulose. So these are two important terms. These are two more disaccharides. What are the properties of oligosaccharides? They are again sweet to taste. They form true solution in water, but they diffuse slowly through cell membrane because monosaccharides they are very small sized molecules. Oligo they are formed by the union of 2 to 6 monosaccharide units, that's why they diffuse comparatively slowly. What are the functions of oligosaccharides? They are important source of fuel. Disaccharides they act as source of fuel. They are storage products as we have studied sucrose which is the storage product of sugarcane and sugar beet. Then regarding the cellular interaction, oligosaccharides which are attached to cell membrane, they help to recognize cells of their own kind. So these are the important functions of oligosaccharides. With this we finish the oligosaccharides.